Hello and welcome to this webinar titled SUS Leachable Testing, Leachable Study Design for Single-Use Components, hosted by Biopharma Asia Magazine, and presented by Kathryn McGohan, Dr. Hélène Pora, and Dr. James Hathcock. My name is Sri Patel and I will be the moderator. But before we begin, I would just like to remind our viewers that this event will be hosting a live question and answer session at the end of the presentation. You can submit a question at any time throughout the presentation via the Questions tab, located at the bottom of your screen, and we will go through these at the end of the presentation. Now, please allow me to introduce you to our presenters. Kathryn McGohan is a scientist within the Materials Science Group of the Global Manufacturing Sciences and Technology Organization at Bristol-Myers Squibb. Kathryn is responsible for the selection, qualification, and validation of single-use products for use in late-stage development and commercial biopharmaceutical manufacturing processes. Kathryn also supports the authorship of CMC regulatory filing sections, new product launches, and continuous improvement efforts. Kathryn currently represents Bristol-Myers Squibb on the Bioforum Operations Group, Disposables Workstream. Dr. Hélène Pora is Vice President of Single-Use Technology within Pall Life Sciences, where she has the global responsibility for the marketing and development of Pall single-use technologies. Hélène has been instrumental in the development of Pall single-use technologies for the last 20 years. She is as well heavily involved in quality and regulatory aspects of single-use technology. Hélène has over 30 years of experience working for the biopharmaceutical industry, the last 26 years within Pall Corporation. She speaks regularly at conferences about single-use technology with strong focus on validation and overall process integration aspects. She is well involved in different working groups in the industry involved in standards development, as in on the board of the BPOG and the SUTAP. Dr. James Hathcock is the Senior Director of the Regulatory and Validation Consulting Team at Pall Life Sciences, which includes responsibility for the Extractables and Leachables characterization strategy to support the safe and successful end-user implementation of technologies enabling pharmaceutical manufacturing. He is an active member of the ASTM, PDA, ISDE, BPSA, and the USP 665 expert panel. Since joining Pall in 2008, James has led chemical and performance characterization of medical and biopharmaceutical products, as well as relevant technical packages supporting regulatory filings. Prior to joining Pall, James was a professor of hematology at the Mount Sinai School of Medicine in New York City, where he served as the director of the Protein Purification Core Laboratory. I would now like to invite our presenter for the first half of this webinar, Kathryn McGohan, to join our webinar. Welcome, Kathryn.
Thank you, Sri. I'd like to thank the organizers for the opportunity to speak today, and thank you to the audience for attending. So as Sri mentioned, I'm going to talk about single-use systems, leachable study design for single-use components. Just to give you a quick overview of what the presentation will include, I'll go through extractable and leachable definition and sources, the regulatory background, how the BPOG team has worked to develop a best practice guide for industry and how we've implemented that. Includes a risk assessment, a leachable study design, and analytical methods component. And then I'll go through two case studies. One is a case study where we have a bioprocess bag and another with a filter. And I'll go through those applications, the study designs, and the associated results. First, I'll give you the definitions that I'll be using for extractable and leachables. Extractables are chemical entities that can be extracted from a consumable into a solvent under controlled conditions that are usually more aggressive than normal operating conditions. So those can be conditions of time or temperature or chemical characteristics of the solution used. Leachables are chemical entities that are contributed to a solution from a consumable that happens under normal processing conditions within our manufacturing process. So as you can see in the diagram, typically leachables are a subset of extractables, but it is possible to see leachables that do not show up in an extractables profile. Now I'll go through quickly the regulatory background for extractables and leachables. So for extractables, there are no distinct regulatory requirements to govern industry approaches to extractables. The extractables data packages available from single-use component suppliers are variable and often difficult to compare. And for leachables, 21 CFR states that equipment used shall be constructed so that surfaces are not reactive, additive, or absorptive, so as to alter the safety, strength, or purity of the drug product. And in BPOG user experience, health authorities have variable approaches and variable expectations for what biopharmaceutical manufacturers will be able to provide in terms of leachables assessment and qualification packages. So the BPOG team the Leachables team was formed in order to drive standardization for leachables. I should mention that prior to the Leachables team forming, there was an Extractables team that put out the BPOG Extractables protocol. So that was done first, and then the Leachables team was formed. And so once the Leachables team was formed, the goal was to develop a standard approach to leachables as well. Our first step was to go through a benchmarking phase. Team members representing 22 biopharma manufacturers got together to share information around what are our current practices, what sort of interactions we had with health authorities, and various inspection outcomes that we were able to share with one another. And so we looked at those and we were able to define our scope, and we decided that we would focus on 3 specific areas. Those included the risk assessment, the study design, and the analytical methodology sub-teams that we formed. So I'll quickly go through the risk assessment best practices that the team, over a period of, I think, about 1.5 years, we were able to come up with. So how do we define extractable and leachable risk? So the risk assessment allows us to perform a comprehensive risk assessment using defined criteria for each consumable used at every process step in our manufacturing processes. The criteria that the team ended on was the process step utilization, or that's also called the distance along the production stream, and that's basically looking at where a particular consumable is used within a manufacturing process. The second consideration is solution characteristics. So if you're using a neutral aqueous solution, or maybe you're using a caustic, that will be taken into account during the risk assessment. The exposure duration and temperature are sort of self-explanatory. How long will the component be in contact with a particular solution and at what temperature? And then the surface area to volume ratio is the final consideration for the risk assessment model. And that looks at what is the surface area of that single-use component and what volume of solution will be processed through it or held in it, depending on the use of the component. So this is sort of a hard table to look at, but it basically breaks down those criteria that I just went through into various categories based on those criteria. So, for example, the distance along the production stream, something that's used in synthesis or cell culture would be considered to be lower risk than something that's used in the final formulation or fill finish. For exposure temperature, the warmer the temperature, the higher the risk, so it would receive a higher risk score. Exposure duration, longer duration gives more time for chemical reaction to happen, so longer duration is scored more with a higher risk rating than a short duration. The process fluid interaction, that's those chemical characteristics that I spoke about before. So as you can see, a neutral solution would be a lower risk than a surfactant or a polar solvent or a solution that's highly concentrated. And then the dilution ratio is the measure of the surface area to volume ratio. And again, that's based on the surface area of the component that's in contact with the stream and the volume of the stream. So the lower the volume in relation to the surface area, the higher the risk there would be. So again, this is just another way to look at that risk rating. So we score each consumable and process stream in our manufacturing process according to those criteria. In the BPOG guide, it allows for each company to determine their own criteria and weighting. So based on that individual company's knowledge or experience, they can weight each of those criteria accordingly. So you take your scores for each of the criteria, multiply by your weights, and add those up to get a final Leachables risk rating. So the possible range for Leachables risk rating is from 1 to 9. So we broke those down into low risk, medium risk, and high risk. So anything that scores higher than a 6.3 is considered to be high risk. So the next step would be risk control once you've identified your risks. So consumable and process stream interactions with a Leachables risk rating of 6.3 or higher require additional evaluation or control. One way that we're able to mitigate that risk is to use existing data, whether that's data that's been provided us through a supplier, or whether we've performed a previous study with that component in a similar solution. We may be able to leverage that for the current usage. If that's not possible, then we need to move forward into either a controlled extractable and or a leachable study. So I mentioned that we're able to use supplier data in some cases to mitigate the risk. So when we look at supplier data, we'll evaluate whether that data comes from the BPOG protocol that I mentioned before. That's a standardized extractables protocol that the BPOG Extractables team put out in 2014. That includes 6 solvents and multiple time points, and that varies by the type of component. That's something that we ask for in our statement of user requirements when we are sourcing a component. We'll ask for this type of data. Sometimes that data is not available. When that's the case, we'll review what data the supplier can provide us. And on a case-by-case basis, we can determine whether or not it's sufficient to cover the application of concern. If it's sufficient and we have identity and quantity of compounds that are extracted from a particular component, then we'll require a toxicology assessment in order to qualify a component for use. When the supplier data is not sufficient for us to leverage, then we may look at what BMS already owns. So we may have a study that was done for a different program. We'll look at whether or not the appropriate study design for that application exists, whether the test solution brackets the solution that we're looking at, whether the exposure time that was done in that study is sufficient to cover our worst case conditions for the process step of interest, and whether or not our extractable and leachable compounds were identified and quantified. That identity and quantification is critical because without identity and quantity, we're not able to perform a toxicology assessment to look at the risk to patients. So now I'll go through the Leachable Study Design and analytical methods portion. So if we decide that we're going to do an extractable study in-house, then we will look at utilizing the BPOG solvents and time points. In some cases, we may modify that, but we would carefully consider what our potential future usage for that component is, because whenever we do a study, we want to ensure that we're getting the maximum data from that study and that we can minimize having to do rework. So in that study, we would obviously want to identify and quantify any extractable compounds. We would perform a tox assessment if necessary. It's necessary if there is anything in the extractables profile, we need to look at that. So the way that we do that at BMS, we assume that the full amount of an extractable that's detected is forward processed into 1 dose. If we have an unknown compound, then we would utilize a threshold of toxicological concern approach. And that's assuming that we assume that that unknown is a very toxic compound through that approach. If a compound of concern is detected in an extractable study, and that means something that is either an unknown or an extractable that is in a level that we're not comfortable with, we would move forward into a leachable study. When we design our leachable study, we want to ensure that the leachable study is efficient. Similar to what I said about the extractable study, we want to ensure that when we perform our study, it will cover the full range of processing conditions, including that exposure duration, temperature, and our surface area to volume ratio. We use the best practice analytical methods that are outlined in the BPOG guide, and doing this helps us to reduce rework and any validation gaps. So this is an overview of the basics for setting up a study. So we would look at the component sample selection. In most cases, we would use a scaled down approach. If we're using, for example, a 30-inch filter, we would perform testing on a much smaller filter in order to be able to use a amount of solution that allows us to get the best results and have it be comparable. So we would also look at pretreatment. If a component is gamma irradiated, we would want to ensure that our test sample is gamma irradiated. We want to look at our contact solution and our negative control. The contact solution should ideally be the actual process solution that we're looking to qualify. If we have to use a surrogate solution, then we would have appropriate justification for that. The negative control should be a solution that's pulled from the original pool of the contact solution so that we make sure that we don't have any false results that are due to just being a different aliquot of the solution. Sample handling in terms of what the samples are held in, whether it's glass or Teflon. This is outlined in the BPOG guide in more detail. Extraction mode, whether it's dynamic or static. If it's dynamic, something like having the solution in a bag on a rocker table, constantly moving, versus just stored static. Exposure conditions, again, the duration and the temperature need to be appropriate for the process stream and consumable interaction that we're attempting to qualify. So the study methods that we would use for extractable and leachable study methods, we would look for organic compounds using GCMS, direct injection for semi-volatiles, and headspace for volatiles. That's suitable to screen for plasticizers, aromatic and non-aromatics, and residual solvents. We'd use LCMS to look for non-volatiles, and that can help us to screen for fillers, plasticizers, antioxidants, catalysts, slip agents, and other polymer additives. And then for metals and elemental impurities, we would use inductively coupled plasma optical emission spectroscopy. So now I'm going to move into the case studies. So the first case study, I'll go through the risk assessment and the study design. And that case study is a DP mixing bioprocess bag. So I'll go through the application, the risk assessment example, the study design, and the results. In the second study, the case study, I'll talk about a BDS filter, which we qualified initially, and that was done prior to the implementation of our best practices. And then there was a subsequent process change, so I'll go through how we reviewed this application and then the retesting plan and results that came out of that assessment. So for the mixing bag, we had a bioprocess bag that was a 1,000-liter bioprocess bag. The process step is a formulation, mixing, and hold. The exposure temperature is ambient, and the worst case that we determined for exposure duration was that it could be potentially greater than a week. The solution in contact is a protein solution with a neutral pH, and the dilution ratio is 0.09 meters squared per liter. So we ran that through the risk assessment model that I walked through earlier. So you can see that for the distance along the production stream, because this was a drug product application, this was very high risk. Exposure temperature was ambient, so that put us at 5. The duration was longer than a week, so that's high risk, 9. The process fluid interaction was a 5 based on just the characteristics of that process stream. And then again, the solution, or the surface area to volume ratio, gave us a score of 5. So when we ran that through the model, the final risk rating is 7, so this corresponds to high risk. As I mentioned previously, if you have a high risk component, we need to perform additional mitigation for that risk. So the process stream was drug product. The component was a bioprocess bag. The first step was to evaluate what supplier data that we had. In this case, the supplier data was not suitable to bracket the in-process solution. The mixing and hold time for that product had not previously been evaluated, so we determined that we would go forward with a Leachable study. So as we designed the study, we determined that we would use a 1-liter scale-down model, and we would use that because it allowed us to have an appropriate surface area to volume ratio. The contact solutions and controls that we used were the placebo and the drug product. The exposure conditions we selected was 14 days at 30 degrees C. If you remember the process application, this was something that we could hold for greater than a week and at ambient temperature, so we went on the high end of both of those parameters. And we chose to do a static extraction mode. Again, as I mentioned, the analytical methods, we used GCMS, direct injection in headspace, LCMS, and ICP-OES. So when the results came back for the study, we had 4 organic compounds that were detected and 3 metals. So that information about the identity of those compounds and the quantity that was detected was forwarded to our toxicology group, and they were able to take the dosage of the product and look at the quantity and identity of those compounds and determine that based on the identity and quantity of the leachables detected in the study, that there was no impact to patient safety based on that. Therefore, these bioprocess bags were appropriate for up to 14 days in contact with that product at ambient temperature. So I'll move on to the second case study now. So this is a drug substance filtration application. I'll go initially through the study design and results for the initial application, and then, as I mentioned, there was a process change, in this case a technical transfer, and therefore a scale up. So at that time we did a reevaluation of risk and process parameters, and then we redesigned our study and we had additional results. So I'll go through each of those. So this is a final BDS filter. It's a polyether sulfone filter. It's processing a protein solution with neutral pH. The exposure temperature is ambient, and the maximum duration possible for this filtration initially was 12 hours. So this was run through our risk model that we were using at the time. It was identified as high risk, and we did not have existing supplier data or BMS zone data that we could mitigate this risk with, so we required a leachable study. So when the initial study was done, the way that it was performed was a BDS sample was taken directly from the manufacturing process stream after contact with the filter under actual manufacturing conditions. And the control sample for this study was taken from the BDS pool pre-filtration. Both the control and the sample were run through the analyses that I've mentioned previously. Again, that's GCMS, direct and headspace, LCMS, and ICP-OES. The test sample was compared to the control, and it was determined that there were no volatile, semi-volatile, or non-volatile organic compounds detected in the stream, and also there were no metals detected. So therefore, the filters were deemed appropriate for use. Then there was a decision that this process would be technical transfer, and as a result, it would be scaled up. Therefore, we needed to look at this application again to ensure that the data that we already owned was sufficient to cover it. So again, it was the final BDS filter. The protein solution was not changing. The exposure temperature was still ambient. However, the new exposure duration was 24 hours maximum. As a result of that change in the time, we looked again at the data that we owned. So again, this is a product BDS. The filter is a PES filter. We previously had a successful evaluation. However, the samples from that previous study were taken directly from the manufacturing process. Therefore, they're representative of the actual manufacturing conditions in the initial facility, but additional evaluation was recommended at that time to represent the worst case conditions possible in manufacturing at the scaled up process parameters. So additional testing was required at that point. So we went through determination of worst case for the application in speaking with the process engineers and several folks, we determined what the longest potential exposure was, and we used that to define the conditions for the study. And in this case, we knew that the in-process filter is flushed. So in order to make this a really worst-case study, we've eliminated that flush. It is possible that you could run a similar study and do a flush. It was just our decision at that time to eliminate that flush so that we could get the worst-case sort of leachables profile. So the BDS test sample was taken from the BDS pool pre-filtration. 1 aliquot of that sample was reserved as a control sample, so it had no contact with the filter. The other aliquot was placed in contact with the filter for a period of 24 hours at 25 C, again with no filter flush. So we used the same analytical methods and conditions used in the initial studies, and we were able to detect 3 organic compounds and 1 metal compound in the BDS. So as is our normal process, when we have any leachable compound, we look for the identity and the quantity. We pass that on to our toxicology colleagues, and they're able to perform an assessment that's based on the product dosage to the patient, as well as the quantity that was seen in the stream. Again, they looked at this and were able to determine that based on the identity and quantity of the leachables that were detected, there's no impact to patient safety. Therefore, these filters are considered appropriate for use now up to 24 hours in contact with the BDS at ambient temperature. So in conclusion, I just want to go back through. So the BPOG Leachables Best Practice Guide is available. You can look through it for a much more detailed view of the risk assessment model. That model provides a framework for consistent, efficient, and robust risk assessment of consumables used in a biopharmaceutical manufacturing process. It also provides more detailed information on the Leachable Study Design, the study parameters that should be considered in order to adequately support our worst-case processing conditions. And then our case studies, I hope, showed that a strong process understanding is required for a well-designed leachable study. When you're going to qualify something, you really need to understand your usage and how you can best support that in order to ensure that you're assessing the current and probable future applications. That way you can eliminate future rework. So the overall process for BMS, for E&L, is we start with a risk assessment. We do a robust study design and data analysis. Then there's a toxicology assessment, and at the end, our goal is that we'll have improved patient safety. We want to ensure that, you know, we're not having any impact to our patient safety. So that's the goal of this this program here. And with that, I will hand it back over to Shri. Thank you all for listening.
Thank you very much, Kathryn. So just as a reminder to our listeners, if you have any questions for any of our presenters, then please click on the questions tab and send them through to us, and we'll be asking these at the end of the presentation. So I would now like to invite the speakers for the second half of this presentation, Dr. Hélène Pora and Dr. James Hathcock, to join our webinar. And during this part of the presentation, there will be some survey questions which we'll make active in the voting tab, and we'll make them active during certain points during this part of the section, the webinar. So welcome, Dr. Pora and Dr. Hathcock.
Okay, thank you very much, Shri, for the nice introduction, and I have to say that Kathy's presentation really paved very well the road for what we want to discuss. And I think really, I mean, the focus of what we are going to discuss is really looking at how, as a supplier, we can provide data that will really help the end users to really fulfill their risk assessment and then decide on an adequate basis whether any additional testing is requested. So on the next slide, I mean, I really want to ask a question to understand really where the audience is coming from and what is their interest related to single-use technology, and particularly whether they have a new project that they want to implement next year. So Shri, I mean, we left about 30 seconds to the audience to answer the question then.
Okay, I've just made the voting live now, so it's going to be live for around a minute. If you could vote for the answer you like. Just to let you know, just click on the voting tab and make your vote. Give it another 10 to 15 seconds.
Yes, another 10 seconds, and then we move forward. Okay, I suggest we move forward, Tree.
Okay, go ahead. I'll end the voting there.
Okay, thank you. So really, I mean, if I look at what we are going to discuss, we are first going to discuss the ownership of single-use material. Then we will discuss our approach about how we have been implementing and how we are still implementing BPOG, I would say, testing recommendations. One of the very important points is that we go through what are the lessons learned from a technical point of view, and as most of you are aware, there is as well the discussion related to USP 665, so somewhat about, you know, what does it bring to the table. And then we will discuss the remaining challenges, because as you can imagine, as your guests all quite active in this industry, there are still a few ones which are staying ahead of us and which will keep you busy for still some time. So I think that's one of the important points, and that's the reason why I would say there is a lot of communication to that subject between end users and suppliers, is because at the end of the day, when we speak about single use, what we speak about is really how do we share the risk, and basically how do we share the responsibility. And what is clear is single use has some kind of implied that there is a shift in terms of a shared responsibility from what was in the past at the end user side and moving to the supplier side, which does mean that I would say there is increased responsibility, and then we have to be respectful for this and really ensure we are doing what needs to be done for our end users. So basically, I mean, the concern around extractable and leachable has been perceived as a concern, this for a long time, and when you look at the results of studies, kind of year after year, you don't see, I would say, a lot of change, which still means that it's an active subject we all have to work on. And basically, that's why as a company, we have put a lot of efforts, you know, related to addressing these concerns and helping the end users. So with this, I would like to move to my colleague and friend, James Hathcock, who will take you to the work that we've been doing for quite a long time.
Excellent. Thank you, Sri, Katie, and Hélène. So as Hélène pointed out, the concern in the industry for extractables has been quite evident and quite at the top of the list for a number of years. And when we think about a multi-component extractables E&L risk evaluation, right, it starts with the product and the process. And then when you get to the single-use system, often in the past, the approach that's most cost-effective for an end-user risk assessment is to focus on the entire ensemble single-use system with specific model solvents that represent the product and process to generate the data. However, we know that if every end user does this for every single-use system and every specific process out there, there's a lot of redundant testing going on in the industry and a lot of wasted testing effort. So the proposal to move forward with component-specific testing is that manufacturers will understand and better characterize the components that they provide in a way that hopefully brackets 80% of applications. And then the data for each of these individual components can be put together in an ensemble approach to help address the initial on-paper overall extractables risk assessment, right? So in a high-level view, the table on the right shows if you put together the individual data sets from suppliers on each of the components within a single-use system, this is without any testing, maybe you rank them by abundance or the highest surface area component to least surface area component. You indicate which solvents for which you have data available, and then you add up the worst case levels of compounds from each of the components to get a total sum or ensemble levels of compounds for the entire system. You would then couple this in the middle section in blue with process-specific and application knowledge that the end user knows, such as batch size, product concentration, maximum dosing per day, to come up with an upper bound estimate of the worst case levels of each individual compound that could be exposed to a patient per day. And then in addition, you couple that with toxicology information shown in the bottom table on the right to understand whether those worst case levels need to be considered for toxicology assessment, maybe coupled with PDE calculations and so forth. So that's the high-level approach, and it's intended for your initial risk assessment. How can you do all of this as an on-paper exercise, minimizing any testing needs? So to do this, to be able to pull data together from different components into this ensemble system approach, standardized data, as Katie mentioned, is very helpful. So the benefits of having standardized data in a way that meets and aligns with industry groups and expectations are that we hope it facilitates and drives the adoption of single-use technology. It also generates cohesive and reliable extractables data sets for both suppliers as well as end users. And then lastly, with regard to the industry as a whole, we expect it minimizes the end user testing requirements. So in late 2014, the BPOG end user group published an extractables protocol intended to be used as a user requirement specification. Right? And this is very specific for different types of single-use components, which solvent portfolio is required, which time points, temperatures, etc., would be expected for a supplier to be able to provide that really minimizes the testing needed for each individual component and application. So this approach was very resource intensive, with 6 solvents, 3 to 4 time points per component. So at Pall, we decided we wanted to implement this approach. And so it starts by going in and understanding what resins are used in manufacturing, where they're used, coming up with component families. So what filter component would we want to test to represent the most part numbers that we provide? Then we looked at the method optimization. So although Pall has performed mass spec analysis of extractables since the mid to late 1990s, technology has evolved, and we wanted to make sure that we built a library that well characterized the resins and the components that we used. Part of this meant that we needed to create a stronger library of what compounds we typically expect to see from our resins. And really the goal is first perform high resolution or accurate mass spec screening of characteristic components. And then based on that library information, the aim was that we would understand our materials and components better than anyone else before any end users ever saw compounds in their data, right? So now moving forward with the actual testing, here's what you see a typical BPOG extractables report from Pall contains. And so the first point is technical reports would be different for an autoclave component versus a gamma irradiated component. Although some components can be sterilized either method, the profiles may be slightly different and warrant different reports. The reports also have a descriptive summary of the major observations, a table taken from the BPOG ISPE paper outlining the extraction parameters that were performed, the analytical method details, as well as images of how the extraction was done. Right? Then in the lower left, if we explode the view of the grand summary of the organic extractables table, what this does is show for each solvent evaluated, the compounds observed ranked from most abundant compound to least abundant compound for each solvent. We also describe the compounds used or how these compounds were semi-quantified, so what was the reference compound, as well as the LODs, LOQs, and so forth for each compound. If we say we did not detect any compounds by a certain method, we show that in gray, but we're very clear to state what compounds or reference standards were used to say no compounds were detected below this limit. So similarly, for the elemental impurities table, we started with all ICH-Q3D elemental impurities, which is required as part of a risk assessment in the US and Europe, effective in January. And these are ranked from ICH class 1 to class 3 for the elemental impurities for simplicity. We have a reporting limit of 20 ppb is required, and then also include other elemental impurities that were easy to include in the screening portfolio as well. So that was talking about the data. In the appendixes to each of these reports, you also have a list of Pall part numbers and relevant surface areas for the materials to enable you to easily scale the data to larger or smaller size components within the same component family. We'll also walk through an example of how a typically scaling could be performed. In addition, we'll also have references to typical toxicology information such as ICH Q3C for residual solvents, Q3D elemental impurities, or in some cases, we're building a library for the most abundant compounds observed based on model applications having PDE calculations for your risk assessment. So where are all these reports stored? Of course, they're stored in our quality database. So the same place where you pull up any specific Pall part number, you see statements for USP 661, USP 87, 88. In the exact same location, you'll find the always current state of the BPOG testing reports that are available. In terms of status to date on our BPOG extractables testing effort, we've had a dedicated headcount in place towards focused on this testing since 2015. It's now a standardized Pall requirement for this approach for all new component development, as well as for any material changes where we already have existing data. We're also working with sub-suppliers, those manufacturers who provide sensors, tubing, and other components that Pall doesn't manufacture, but which we integrate into single-use systems. So working within the community so we can all have in-kind data sets to help conduct this ensemble approach. Right. On the right-hand side, you see a table of Pall manufactured components at the top, listing testing that has either been completed to date or that is currently testing in progress and expected to complete by the end of this calendar year. So we have a large number of reports available to date, and we expect it to continue growing throughout the calendar year. Okay. So this takes us to our second survey question, and I think Sri will help pop up the survey poll to make it available to you. And the question is, how do you perceive Pall's BPOG, extensive BPOG component alignment program to help facilitate single-use adoption?
Okay, everyone.
30 or 40. Yeah.
I've just made the voting. Oops. Yeah, it's now. Yeah, the voting is now live, and you can make your vote by the voting tab. And just to let the audience know, the voting is now live. So please make your selection by clicking on the voting tab. It's going to be open for another 20 to 30 seconds. Okay, just another 10 seconds. Okay, great. I'm going to end the voting there. And back to you, James.
Excellent. So thank you, everyone. So going through implementation of the protocol, generating a fair number of reports to date, you could imagine that we've had a number of lessons learned, things that worked really, really well, things that worked kind of well, but we needed to learn a few tricks and so forth, and things where we had challenges that needed overcoming. So in December of 2017, a few months back, we published the first of a two-part article in BPI talking about experiences learned during the implementation of the Extractables protocol, as well as the analytical measurements performed on the samples. So that article, you'll see a reference in the bottom left where you can find it online. But basically, we talk about, can we achieve the surface area to volume ratios that are expected or desired, how we did that, how we performed the mixing or agitation, and if we could not achieve those surface area to volumes, what did we feel we reasonably were able to accomplish and how we reported that. We show images of how we approach biocontainers, aseptic connectors, and sterilizing grade filters, and also talk about some of the challenges with the instrumentation and basically how we restructured our approaches slightly to best mitigate these challenges. So that article you can find by the link on the bottom left. Along the same time, Hélène mentioned that although BPOG is an end-user requirement that was published at the end of 2014, there's been a lot of active discussion from USP chapters 661 to 665 about coming up with a standardized approach for supplier extractables data sets to help qualify or to help describe and characterize components. So some of the key highlights and differences between the BPOG and USP protocols, again, one's a finalized end user requirement that's published and readily downloadable. The other is a draft standard that's going to go out for another round of public comment this year. The BPOG protocol is 6 solvents with 3 to 4 time points, so very resource intensive, but tends to fully bracket a vast majority of applications, whereas the USP 665 approach tends to minimize the number of extractions and time points in a way that well characterizes components that may not fully bracket as many application conditions. Okay. So we talked about lessons learned from implementing BPOG. We've also run some studies using the USP 665 draft protocol that was published in May of 2017. So recently in February of this year, we published a second BPI article. Again, you see the link on the bottom left. And here we talk about some of the key differences in the two protocols and whether they appear to have an impact on the extractables profile that you see. So specifically, some of the graphs show chromatograms observed using the BPOG pH 1.6 solvent as compared to the USP pH 3 solvent for the low pH extractions. And very similarly, we've also looked at pH 10 and the pH 13.5 high pH solvents as well. And for the most part, with the polyethersulfone filters, we saw very strong similarities in the profile. There are some subtle differences, tend to be at low levels. And once you understand what the identification is of those compounds, then it's more clear what the scientific story behind those differences may be. Right? So this article is available online via the link below. Okay. And lastly, I want to come to where are we now as an industry with the BPOG protocols and some of the challenges that I think the industry will focus on and where we'll get much better by the end of this calendar year. Right? So some of the questions we expect to see are who's performing BPOG, who's performing USP. As an end user, will I be expected to have both data sets available? Right? And the only comment I can make here is that there's strong interest from both teams to ensure that the protocols are well aligned. Certainly for the low pH protocol, USP has some comments in the draft proposal saying that there appear to be similarities between the BPOG low pH solvent and the USP recommended solvent. The 50% ethanol extraction is the match to both protocols, at least at the single time point. And the high pH conditions are whether there's a little more discussion ongoing about whether pH 10 and 13.5 generate the same profiles. And I think the jury's still out, but there's certainly a strong interest to ensuring that two protocols are not required. Okay. Similarly, there's an ongoing issue that'll be the focus this year in terms of component supplier transparency. So very often different suppliers may have, some are generating the full BPOG protocol, some are generating a subset of the BPOG protocol, some are waiting to see what happens with USP and whether that should be the future approach. Other suppliers may have their own individual approach, and then thirdly, others are simply waiting and not generating anything at the moment and will change maybe in a year or two. We talked about sharing data, so once you understand who's generated what data sets and what's available in the industry, there are also some challenges in terms of how we as integrators can pull together these data sets and be able to share these to end users with the single-use systems. Some of these challenges are, is a three-way confidentiality agreement required? For some component manufacturers, that's the case. In other cases, they may ask the end user to contact the component manufacturer directly. So it's not quite as easy as we would like to pull together all the component data within a single-use system. And in other cases, they may say, yeah, we can share the data. You can share the data with the end user, but make sure I know the customer's name and contact information, right? And in a world where sub-suppliers and integrators are sometimes friends and sometimes competitors, it certainly says we need some ground rules to be generated this year, and I think that will be a focus of BPSA and BPOG activities for 2018. One last comment on the alignment between the BPOG and supplier community is coming up with a standardized template for reporting the data. Certainly a draft template was published a number of years ago. I think a lot of end users have sought to optimize this for their specific needs, what makes sense. But as a result, we sometimes have a number of different Excel-based templates from different end users. And I think as a community this year will be a great opportunity to just update, improve, but have one Excel-based template for reporting data at the end of the year. Okay. So the last category is missing data, and Katie mentioned missing data a bit as well. So I think as suppliers focused on generating these data for the most high-risk components or components used in high-risk applications, certainly with more than 2,000 components on the radar, you have to prioritize those that would have the most impact for the end user first, and then gradually over the next 2 to 3 years, get to the other components in your portfolio. So as an example of this, in 2014, before the publication of the BPOG ISPE protocol article, we had generated data on a single-use TFF system and flow path. So you have the TFF module, but you also have tubings and fittings and connectors that really allow you to easily use this system in a process application. And to help support this, we had provided some generic extractable studies on the entire system flow path, which contained about 15 different single-use components within that flow path. And then at a later date, there was a new sensor was identified that was incorporated into the flow path. It really helped with the performance and the process monitoring of the system, but we didn't have the same level of data. So this created a data gap in the portfolio characterizing the system. So over the past 6 months to a year, we've had a number of interactions with this sensor supplier. It's been a good story in the end. The sensor supplier is now generating not the full BPOG protocol, but at least a 50% ethanol extractables data set that at least covers one specific application condition. Right? And they're also aware and anxious as the BPOG and USP come on board to complete a more robust study portfolio. Right? So that was one specific example. But also, a lot of the focus in the industry has focused on what we do for components in high-risk applications, whether it's the USP protocol or whether it's the BPOG protocol. The approach to identify the specific compounds, levels, and how you do a toxicologic risk assessment has been a primary focus the past few years. As testing resources in all labs are going to be limited in the next few years as these everyone begins to test their components, the low and medium risk categories have not gotten as much attention. So some of the challenges I think you and suppliers will face over the next 2 years is for small surface area components, what level of data can suppliers provide that will help close the gap for this year and next year, with the understanding that 2 to 3 years down the road, we'll have stronger USP or BPOG data sets on these smaller or lower priority components. So the BPOG approach gives examples of types of data. USP gave a draft version last May, but I think these are still open for discussion. And often these do cause a big pain point on a day-to-day basis. So some of the proposals are USP Class 6, European Pharmacopoeia, the old USP 661, which has now been withdrawn in favor of 661.1, .2, and 665. But we're very anxious to get end user feedback on whether the old 661 approach is a consistent approach that will meet the gap in the next few years, or NVR, TOC, and so forth. And I think as Katie indicated earlier, typically these are all done on a case-by-case basis, but it would be great to have some industry alignment here. Okay, so we've covered a number of the successes that we've had, as well as ongoing challenges with implementation of single-use data sets. We've talked about the materials ownership, the implementation strategy for the protocol at Pall, some of the lessons learned and where you can download the articles that tell more about the learnings and experiences in the past year, as well as what are some remaining challenges we face that we hope to get to a good place on by the end of 2018 as a supplier and end user community. So that's it. So that takes us to the third and last survey question that we have. And Sri, if you will pop this up and I'll read it aloud, which is, do you think that Pall's E&L BPOG data and experience would benefit you with your implementation of single use?
Great, and I've now made that live, so please click on the Vote tab and please make your selection. Okay, and just let the audience know, click on the voting tab and you'll see this voting question there. And you can make your selection or answer. Give it another 20 seconds. Okay, thanks very much. I'm going to end the voting there. Okay, that's the end of the voting for that question. James, thank you very much. Hélène, thank you very much. We will now begin the question and answer session. And just as a reminder to the audience, please click on the questions tab and send any questions through to us. Let us know if there's anyone specifically you would like to ask. Other than that, let's begin. So the first question, which extractables testing approach will the industry eventually gravitate towards? BPOG, USP, or both? That's for everybody.
Yeah. So, James, I'll take this one. Maybe good to hear from Katie as well. But I think the good news is, right now, the industry knows we need standardized approaches and consensus data that makes it easy to assemble the story. Right now, the BPOG is out, it's published. It's been published since late 2014. and we know there's another round of public comment on the USP draft proposal. We know that both groups, BPOG members as well as USP members, want strong alignment so there's not redundant testing to have the implementation of a new standard. But Paul's approach today is we're going with the standard, the user requirement approach that's been published, that's out there, that meets end users' need today. And then what I think you will see is you'll gradually see alignment and a consensus position over the next 2 years where we have one approach that largely fits both needs.
And Katie, if you'd like to answer that as well, I'll just give the rest of the question again. Which extractables testing approach will the industry eventually gravitate towards? BPOG, USP, or both?
I think that, as James mentioned, all of the above organizations are working hard to come up with something that's going to work for everyone. So I think that we will see additional collaboration between the groups, and eventually there will be something uniform that we can settle on.
Great. Next question. How does the supplier provided extractables data fit into the Leachables assessment program?
Okay, so I can take this one. So the supplier provided extractables data is our sort of first step in looking at any component that we're looking to use in a process. So we will, when we've done our assessment, we'll look at that, and we can use that to target specific leachables if there are things in the extractables profile of concern. Or as I mentioned before, we may be able to perform a toxicology assessment on the extractables that are present and use that in order to bridge the gap that we have in our qualification. So the supplier data is really our first stop when we're trying to mitigate the risk of any particular consumable and process stream interaction.
Great, thanks very much. The next question, what are the options for extractable risk assessment when complete set data for components are not available?
So I'll talk first, and then maybe Katie can chip in as well. So as Katie indicated, these are often a very much case-by-case basis. Certainly the BPOG extractables and leach approach tells you how to identify a component that falls into a low-risk application, so we know it fits into the low-risk application, but what data gets you through the day. From our approach, often this has been the traditional USP 661 data, which has a NVR value. Some end users are using TOC values on these approaches. And then in some cases, you may have a single compound-specific extractables profile in either water or 50% ethanol at a single time point. And that's been our approach, and we'd love to see some more standardization and consistency here as well. But good question.
Okay, great. And would anybody like to add to that, or shall I move on? Right, I'll move on to the next one. So the next question, how do you set acceptance criteria for the results from a leachable study?
So for the leachable studies, what we do, as I mentioned, when we perform a leachable study, we're looking for both an identity and a quantity of a particular compound that's found in the profile. So really the acceptance criteria is when we take that information and we look at it through the lens of what is the dosage that's going to be delivered to a patient. We're looking at, you know, that compound's permissible daily exposure and things like that. So our toxicology team really is key in this. So they'll look at the data that they have in terms of safety. They'll look at what is the identity and what is the quantity, and whether or not that patient exposure is likely to provide any impact to the patient. So obviously anything that we think has the potential to impact patient safety would be a fail. So it needs to be able to be put through that toxicology assessment successfully in order for us to be able to use that component in that application.
Great, thank you very much. I think the next one is for Katie. You mentioned that you was looking at BMS-owned data. Does BMS have specific extractable and leachable databases for this purpose?
I wouldn't say that we have necessarily a database, but we do have a long history of performing these types of studies, and we have the data that has been generated through all of that experience. In addition, we have many supplier-provided data sets for various components. So when we receive a supplier data set, we'll archive that as well. So we have both, you know, the history of extractables information that we've gotten from several supplier sources, as well as all of the internally performed studies. So we do have a good amount of archived data.
Great, thank you very much. I believe the next one is for Hélène and James. How do you find the extractable and leachable assessments mentioned in this presentation for Pall filters and components?
Yeah. So great question. So the first approach is you've got a Pall representative to reach out to them, and they'll kind of match it with your needs, and they'll reach out and make sure you get the data sets. A second option is Pall has an extensive validation services program where we do not only the BPOG extractables approach, but process-specific extractables testing on components and systems that match your application. You can get that email contact link through our website for Pall Validation Services. They also have a number of other services that are offered as well. And thirdly, as the last option, you can reach out to me and I'll try to steward the question appropriately. But the data sets are readily accessible and we'll make sure you get what you need.
Great, thank you very much. The next question. If extractable is not, then is it required? Oh, I think it means if it's not extractable, then is it required to perform a leachable study?
So I think I can take this one. In general, we do prefer to have extractables data. So it may be that when we identify component interaction that is high risk, we may perform an extractable study first and then move forward into leachables. But sometimes we'll go straight to leachables. It really depends on the what is the application and what are we looking to get from that study. So I would say that certainly in any case where we have high risk, we need some sort of data, and it's case by case whether or not we can use extractables or leachables is required.
Great. Thank you very much. Unfortunately, that's all the time we have today. I would like to take this opportunity to thank our presenters, Kathryn McGohan, Dr. Hélène Pora, and Dr. James Hathcock for sharing their knowledge with us. Audiences can view this webinar on demand by visiting biopharma-asia.com. Thank you very much, everyone.
Thank you.